Welcome to the deep dive. We cut through the noise to bring you the insights from, well, really cutting-edge research.
That's the goal.
And today, we're diving into something in cancer biology that's genuinely thought-provoking. We know cancer cells are, well, incredibly cunning. They adapt, they evolve.
Constantly changing.
Right, finding new ways to escape treatments. And often, we focus on genetic mutations or maybe these complex internal signals driving resilience.
The internal chemistry.
Exactly. But what if there's this powerful, maybe overlooked external force? Today, we're exploring a, frankly, surprising new trigger that's really challenging how we understand cancer's adaptability.
This sounds intriguing.
It is. Our guide today is a really fascinating article from SciTech Daily. It reports on some groundbreaking research, just published in Nature, no less.
A top journal.
Absolutely. Co-led by Dr. Richard White at Ludwig, Oxford, and Dr. Miranda Hunter at Memorial Sloan Kettering. So our mission for you listening is to really grasp how the physical environment around cancer cells can dramatically alter their behavior.
Making them more dangerous.
Yeah, making them more aggressive, more resistant to therapies. Get ready to maybe connect some dots you hadn't considered. How these external forces have these really profound internal effects.
And it's an important point to maybe stress right away. Our standard view of epigenetic changes.
Right, those gene expression controllers.
Exactly. The reversible modifications that control how DNA is expressed without, changing the actual code. That view is mostly centered on chemical signals inside the cell.
Like tags on the DNA.
Precisely. Think of it as this finely tuned internal chemical system. But this new work adds a critical, and I think previously really underappreciated dimension, the role of physical mechanical forces.
Yeah.
And this isn't just like a biological curiosity. It really has significant implications for how we strategize, how we develop future cancer therapies. It could potentially unlock entirely new ways to intervene.
Okay, let's delve into this then. Maybe just a quick refresher for everyone listening, even those familiar with cell biology. Can you just briefly outline what epigenetic changes are and why their sort of dynamic nature is such a challenge in cancer?
Yeah, absolutely. So epigenetics, it's fascinating stuff. These are mechanisms that regulate gene expression, basically turning genes on or off. But unlike genetic mutations, which, change the DNA letters.
The actual sequence.
Right. Epigenetic modifications are more like annotations.
Right.
Or sticky notes on the DNA or on the proteins it wraps around. They influence whether a gene is active or silent.
Okay.
This can involve adding little chemical groups, methyl groups to DNA, acetyl groups to histone proteins, those kinds of things.
Right.
But the key thing, and why they're so tricky in cancer, is how dynamic they are. The reversal, they can be switched on, switched off, turned up, turned down. Much faster than genetic mutations have.
That's more flexible.
Exactly. This flexibility, this plasticity, lets cancer cells adapt really quickly to stresses, like therapies, by just changing their gene expression patterns without needing a whole new mutation.
So trying to target them is like hitting a moving target.
Precisely. It's a huge hurdle for developing effective therapies.
That dynamic adaptability is, what makes cancer so tough. So this new study really shifts things. If the traditional view was mostly those internal chemical triggers, what did White, Hunter, and their team find about the physical conditions around the cells? How did they challenge that old idea?
Well, what they found is genuinely revolutionary, I'd say. The core finding is that physical pressure, like the actual mechanical squeeze from the tissues around the tumor, can act as a direct and potent trigger for these epigenetic changes.
Wow, okay, not just chemical signals, but actual force.
Actual physical force. To see this happening live, they used a really sophisticated zebrafish model of melanoma.
Why zebrafish?
They're transparent. So you can actually watch the tumor cells in real time, inside a living organism. It's perfect for studying how physical forces affect cell behavior in, a complex tissue setting.
Okay, makes sense.
And what they saw was pretty remarkable. Tumor cells that were under tight physical confinement, basically being squished by their environment.
Yeah.
They didn't just sit there. They underwent these dramatic structural and functional shifts.
So they changed shape and behavior.
Completely. They altered their whole cellular program. Instead of just, multiplying rapidly, which is what you expect from aggressive cancer.
Right, dividing like crazy.
Yeah. Instead, they switched on this neuronal invasion program. It basically equipped them to migrate and infiltrate the healthy nerve tissues nearby with alarming efficiency.
So they stopped growing as fast, but became better invaders.
Exactly. It's like a complete strategic pivot driven purely by physical force.
That level of plasticity is incredible. So the cell isn't just reacting to chemicals, it's physically sensing its surroundings. But how does the cell translate that squeeze, that external force, into such a dramatic internal change? Is there like a specific molecule acting as the messenger?
Yeah, you've nailed the core question. And yes, there absolutely is a key molecular player involved. It's a protein called HMGB2,
HMGB2.
right? High mobility group box 2. It's known for its ability to bind to DNA and actually bend it. And this study shows it's central to this whole transformation.
Okay, so how does it work?
What happens apparently is that HMGB2 directly responds to this mechanical stress from confinement. When the cells get squeezed, HMGB2 moves into the nucleus, the cell's control center, and binds to the chromatin.
Chromatin, and that's the DNA packaged with proteins, right?
Exactly, the highly organized complex. And this binding isn't random. It actively reshapes how the genetic material is packaged.
Yeah.
Think of chromatin like a really tightly folded map.
Okay.
HMGB2, under pressure, essentially unfolds or highlights specific regions of that map.
And those regions contain...
Precisely the genes linked to invasive behaviors and, crucially, drug resistance. It makes those genes much more accessible, ready to be switched on.
So the mechanical stress through HMGB2 literally reveals the invasion playbook in the cell's DNA.
That's a great way to put it. The physical squeeze directly exposes the genes needed for invasion and survival.
That's an amazing adaptation. So the cell makes a trade-off. It sacrifices some of that rapid growth
Right, slows down proliferation.
in exchange for becoming more invasive and resistant, all because it's being squeezed. It's like there's a whole chapter in the cancer cell's survival manual just for dealing with environmental stress.
Precisely. And the impact on the cell's fate is huge. Cells with more HMGB2 might divide slower, yes, but they become significantly more invasive and dangerously resistant to standard therapies.
Which makes them harder to kill with chemo.
Exactly. That trade-off is incredibly effective for the cancer's survival. Instead of being an easy target for drugs that hit rapidly dividing cells, they adopt this slower, stealthier, tougher phenotype. It's a strategic shift that makes them incredibly difficult to get rid of. And it really underscores that, targeting only the fast-growing cells might miss a really significant and dangerous group of cancer cells.
So it's not just reprogramming genes. It sounds like these cells also build some kind of, physical shield against that crushing pressure too. How do they protect themselves from just bursting?
That's another brilliant observation, and it really speaks to just how sophisticated this adaptation is. Under that intense pressure, the melanoma cells don't just change their genetic programs. They also significantly remodel their internal skeleton.
The cytoskeleton?
Yeah, the scaffolding that gives the cell its shape and structure. What they do is construct this protective, almost cage-like structure that surrounds and braces their nucleus.
Protecting the control center.
Exactly. And this isn't just passive. It involves a really important molecular bridge called the LINC complex. LINC.
LINC complex. Okay, what does that do?
Think of it like crucial struts or anchors. It directly connects the cell's outer skeleton, which is feeling and pushing back against that external pressure to the delicate membrane around the nucleus, the nuclear envelope.
Ah, so it links the outside stress to the inside protection.
Precisely. Its function is critical. It acts as a shield, protecting the nucleus from potential rupture, from DNA damage that would otherwise happen because of all that squeezing.
Because the nucleus holds the blueprint, including that invasion playbook.
Exactly. Protecting it is absolutely paramount if the cell wants to survive and carry out its mission under duress. It just shows this incredible level of resilience, not just adapting behavior, but physically fortifying themselves against a hostile environment.
Okay, so let's pull back. Big picture time. What does all this mean for how we fight cancer? This feels like it could really change things.
This is where it gets really compelling, I think. This study just profoundly highlights the critical role, maybe an underappreciated role, of the tumor microenvironment.
It's not just scenery.
Not at all. It's an active participant. Its physical cues, the pushing and squeezing, can actually drive cancer cells to reorganize everything: their skeleton, their nucleus, even how their DNA is packaged.
Wow.
And this allows them to rapidly switch states. They can focus on growth when conditions are good, or switch to invasion and survival mode when things get tough, like when they're under pressure.
That switch.
That switch is key. And as Dr. White, one of the lead researchers, put it very clearly, This flexibility poses a major challenge for treatment,
Right.
as therapies targeting rapidly dividing cells may miss those that have transitioned to an invasive, drug-resistant phenotype.
Because the chemo is designed for the fast growers.
Yeah, exactly. If a bunch of cells, under pressure in a tumor, switch to being slow-growing but highly invasive and resistant,
Yeah.
then standard chemo won't touch them. It might even inadvertently select for those more dangerous cells.
Which could explain why some cancers come back or stop responding.
It certainly helps explain it. But, it's not all bad news.
Mhm.
By identifying these specific factors like HMGB2 and the LINC complex that are involved in this critical switch,
We have targets.
we have targets. Researchers are now empowered. They hope they can develop new therapies that could maybe prevent this invasive switch or perhaps even reverse it.
Like a drug to stop HMGB2.
Yeah, imagine a drug that stops HMGB2 from binding to chromatin when the cell is squeezed so the invasion playbook never gets opened. Or maybe something that messes with the LINC complex, making the nucleus vulnerable under pressure.
Making those invasive cells fragile again.
Right. Now, these are still research frontiers, obviously, but understanding the mechanism gives us clear targets for drug development. It offers some real hope for tackling cancer's adaptability.
This deep dive,
Yeah.
it's really given me a new lens on cancer. The idea that just being physically squeezed can reprogram a cell, make it more dangerous. It's both sobering and, well, incredibly insightful.
Yeah, that's quite something.
It's like finding a hidden lever that changes the cancer cell's whole game plan.
And I think if you connect this to the bigger picture, this study just powerfully shows how physical stress is this potent driver, maybe previously really underappreciated, of epigenetic change in cancer.
Yeah.
It profoundly shapes behavior, shapes response to therapy.
It adds a whole new dimension.
It really does. It moves us beyond thinking only about internal chemical signals to properly integrating the impact of these external physical forces.
It definitely changes how we think about the battle, doesn't it? It shows that the environment's push is just as critical as what's going on inside the cell. It really calls for a more, holistic view.
Dynamic, holistic, yes. And that leads to maybe a final provocative thought for you, our listener, to mull over.
Okay.
If external physical forces can so powerfully reprogram cells and influence a disease like cancer, what other maybe subtle environmental pressures or overlooked physical cues might be quietly influencing other diseases, or even just our general health and well-being in ways we haven't even begun to figure out yet?
Huh. Like everyday physical stresses we don't even notice.
Perhaps. How much more is there still to uncover about our body's really intricate kind of hidden responses to the physical world all around us?